First of all, what is synapse? This gap. This gap is called as synapse. Okay, so this gap is said to be synapse. Let's see little bit more focus to be that between the two neurons, this gap is called synapse. Okay, so before the synapse, whatever the neuron is present, that can be termed as presynaptic neuron. And after the synapse, whatever the neuron is present, that is termed as postsynaptic neuron. And very important to be noted down that always the impulse is going to be travel from presynaptic neuron to the postsynaptic neuron only. Because if it is, that is the assertion, what will be the reason? Because only that axon terminals are having the capability to release the neurotransmitters, whatever it is, acetylcholine, sympathin, whatever it is, the neurotransmitter releasing capacity is only here. That means axon terminal. So that area we need to see little bit more. That I have to improvise that size. That only now I am drawing that presynaptic neuron and the neuron of dendrite of other. Okay, like this. So this area, the telodendrites are axon terminals or terminal buttons we can say. Buttons we can say. We'll draw the diagram much bigger. Let me draw the diagram much bigger. Much bigger. So let me show you like this. Earlier I shown this box we are going to draw little bit bigger. So axon terminals are telodendrites that I have to draw. Next, this is the dendrite of another neuron. So let me write here that for our recognition, this is presynaptic neuron. The membrane, we say presynaptic membrane. Okay, this, what do you say? This as postsynaptic membrane. Postsynaptic membrane. The focus here, little bit complicated, but no matter. Once you listen, you people able to write, and moreover, we will get sequence questions like this. So that's why I'm showing clearly. Okay, so these are the synaptic vesicles. So these synaptic vesicles contain a neurotransmitter called acetylcholine. Depends, acetylcholine, sympathin, the norepinephrine, whatever that dopamine, serotonin, various kinds of neurotransmitters are there. We are learning this story with acetylcholine. Imagine that till the time we paid effort in this area, influx of sodium, efflux of potassium, likewise. When the impulse is passing like this, when the impulse is moving forward, that when it reaches here into the axon terminals, when the impulse is reached here, that brings the calcium gated ion channel. Do remember, in this place, what we have? Calcium gated ion channels. Okay, so the calcium gated ion channels open at the presynaptic membrane, and that brings the influx of calcium ions, and calcium ions will come and break the synaptic vesicles. Calcium ion will come and break the synaptic vesicles. So that synaptic vesicles are breaking down, and these synaptic vesicles release neurotransmitter acetylcholine into the synaptic cleft. The space we say synaptic cleft. The release into the synaptic cleft, and that acetylcholine will influence the postsynaptic membrane. That influence postsynaptic membrane. That makes the influx of sodium ions, or that may make the efflux of potassium ion. That any of thing can be happened over. That makes the influx of sodium ion, that make the efflux of potassium ion. I think you know, when if influx of sodium ion occurs, what happens? Charge will increase, isn't it? Then if efflux of potassium ions occurs, what happens? Charge will decrease. Two situations, two different situations we are talking about. If influx of sodium ions occurs, charge will increase. That means it reaches to plus 45. That means we say that is excitatory postsynaptic potential, EPSP. We say that excitatory postsynaptic potential. What is EPSP? excitatory postsynaptic potential, we can say. Okay, I'm writing here excitatory postsynaptic potential. What is IPSP? Charge down, that means go to 90. Inhibitory postsynaptic potential. That means sending information is of two types, isn't it? If you want to say you have to stop the activity or you may raise the activity. That did you remember something earlier we discussed in sympathetic nervous system? Sympathetic nervous system, I said to you that that heartbeat increase, heartbeat decrease, anything can be happened. That means you are sending information to increase the heartbeat. That means you are exciting that area. In excitatory postsynaptic potential, we can say this is postsynaptic membrane. The voltage here called postsynaptic potential plus particle. Then if it is minus 90, what do you say? Inhibitory postsynaptic potential. Excitatory postsynaptic potential and inhibitory postsynaptic potential. Okay. So then a question, a doubt will be raised over. Imagine if in a particular area that two different neurons are sending to different information. One is making the influx of sodium ion, one is making the efflux of potassium ion. Imagine that is happening actually in sympathetic and parasympathetic nervous system aspect when it is making the EPSP and when it is making the IPSP, and both things. One side stop, that means IPSP. One side go. So whom they listen? That will be the question here. Whom they listen? That we can say like that. That depends upon that impulse. Who is more? If EPSPs are more than IPSP, that action will be raised. That means talking about the heartbeat. If IPSPs are more than EPSP, if IPSPs are more than EPSP, the action will get down. Okay. So this is a bigger discussion actually. We have a small statement in the textbook. So that's why I said it over. But at least you people able to understand what is exactly I'm doing here. That the sequence you people need to remember. The sequence. What is the sequence here? Whenever this impulse is moving forward, that will move to the presynaptic neuron. That will move to the presynaptic neuron. Then after that will go to the synaptic cleft by the breakdown of synaptic vesicles. What do you say? That neurotransmitter acetylcholine is getting released over. The neurotransmitter acetylcholine, which makes the changes in the postsynaptic membrane, and postsynaptic membrane will be either excited, we call it as EPSP, or it may get down, we call it as IPSP. So depends upon that action, EPSP or IPSP can be raised over. Okay, so that's it about that. And let us see an animation in this particular area. Let me go for animation. Let me show you animation. Then after we'll discuss back again. Let me share. Ready for you?
The synapse is the connection between two neurons where information is transferred and interpreted. It is a major site for the toxic actions by many insecticidal chemicals. To understand how an insecticide can disrupt the nervous system, it is essential to understand how a synapse functions. Here we see a group of neurons that illustrate a simple network for the interchange of information within the nervous system. Information comes into the network and is distributed between the neurons at their synapse. A synapse consists of a presynaptic neuron that brings in the information signal. The synaptic junction, where the information is transferred from the presynaptic axon to the dendrite of a postsynaptic neuron, and the postsynaptic axon that carries the information away to other neurons. Information is encoded and transferred in the nervous system as action potentials. Action potentials are discrete regions of negative depolarization that are propagated actively along the axon membrane. Let us look closely at the organization of a synapse. The synapse consists of a button-like ending at the tip of the branches at the end of the presynaptic axon. This presynaptic button is separated by a gap from the dendrite of the postsynaptic neuron. The synaptic membrane of the postsynaptic dendrite has numerous sodium channels that are associated with proteins called receptors. Receptors can detect special chemicals released by the presynaptic axon terminal. When an action potential enters the presynaptic axon terminal, the terminal releases a special chemical called a neurotransmitter. The message encoded in the frequency of the presynaptic action potentials is converted by the presynaptic axon into neurotransmitter chemicals that are released and carry the message across the synaptic gap to the postsynaptic dendrite. The neurotransmitter chemicals bind to the receptor proteins associated with the sodium channels of the postsynaptic membrane. Receptor binding of the neurotransmitter chemical stimulates the sodium channels to open and allows positive sodium ions to flow into the postsynaptic dendrite. Transfer of the sodium ions to the inside of the postsynaptic membrane results in the outside of the postsynaptic membrane becoming negatively charged in the region of the open sodium channels. The negative depolarization of the postsynaptic area spreads over the sensitive postsynaptic region and generates new action potentials that are carried away by the postsynaptic axon. To stop the depolarization of the postsynaptic region by the neurotransmitter chemicals, proteins called enzymes are present that destroy the neurotransmitter chemicals. When the neurotransmitter chemicals are eliminated, the postsynaptic sodium channels close. The sodium is transported back to the outside of the postsynaptic membrane by the reactivated sodium pumps, and the outside of the postsynaptic membrane returns to its normal positive charge. It is relevant to note that the synapse illustrated here uses acetylcholine as its neurotransmitter chemical, and the enzyme that destroys acetylcholine is called acetylcholinesterase.
Yeah, this is just information here. Acetylcholine in the synapse, whatever the acetylcholine in the synapse, that should be removed by choline esterase. That should be removed by an enzyme called choline esterase. That means we are talking about the synaptic vesicle. In the synaptic vesicle, whatever that acetylcholine is dropped over, that should be removed, isn't it? That should be removed. That whatever the acetylcholine is dropped over, that should be removed. Okay. So this is the presynaptic membrane. This is postsynaptic. We are talking about that acetylcholine. Okay. So this acetylcholine need to be removed with the help of an enzyme called choline esterase. It's not given in our text actually. Choline esterase. That should remove that area. That choline esterase need to remove that synapse. Synaptic cleft should need to be empty, isn't it? The emptiness is created by that choline esterase. That facilitates that another impulse to come and to release the neurotransmitter. So that is also we call refractory period. If previous acetylcholine is there here, you can't send the second impulse, isn't it? So that's why in the short milliseconds, that acetylcholine need to be removed with the help of that choline esterase. Okay, so choline esterase enzyme removes that. If it is not removed, if you imagine if acetylcholine is still here, if acetylcholine is still present here, then what you can say? We can say the gaps are filled and that's why impulse cannot move forward. That we say that is paralysis, isn't it? That we call paralysis. Okay, so that's it. The other issues we'll get in the coming classes. So just so what I'm saying here, that neurotransmitter sequence you people need to understand. What is the sequence? From the presynaptic neuron, so it will come into the axon terminals. They release the neurotransmitter acetylcholine, which will be received collected by whom? That postsynaptic membrane. Presynaptic membrane, synaptic cleft, then postsynaptic membrane. This is the sequence you people need to understand. Okay. So then, so let me label first of all. So what is this? This is presynaptic neuron. You have to imagine that is presynaptic neuron, axon terminals. Okay. So what is this? This is calcium gated ion channel. That is calcium gated ion channel. And next, what next? This is this area. This area. What is this? That is synaptic cleft. Okay. So then next it is going to be here. Here we have to discuss. What is this? These two. These are said to be the postsynaptic membrane. Postsynaptic membrane. That this is postsynaptic membrane. So what can be expected? Let me show you this also to you. These are synaptic vesicles. All these are said to be synaptic vesicles. So what can be expected in the coming time? So in the animation, when I start the video, the calcium gated channel impulse is coming. When impulse it will come like this, and impulse will come, and let me show you. Okay, so impulse will come that opens the calcium gated ion channel, that brings the calcium ion, that breaks the synaptic vesicle, and vesicles will come nearer and release the neurotransmitter into the synaptic cleft. That makes some changes in the postsynaptic membrane, either EPSP, or we can say, or either they may create IPSP. Okay, either they may create EPSP or IPSP. Okay, let's watch the video. Let me erase all these issues for you. Then we go for video.
To the cell and cause synaptic vesicles to relieve action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
So that's it. This is all about the, either it may create EPSP or IPSP, that you can understand. Okay. So then this is all about what we learned, synaptic transmission. This is what you can say, the synaptic transmission. And